Let's get started with this presentation where I will use metabolomics as a starting point to look into this disease. So what we will discuss today, I'll start with the aims of this project, where I focus specifically on depression. We'll look into data that I selected from the literature dealing with the disease, and then we'll see how the information collected from this data fit very well into what we call an MDD hypothesis, where we make it fit into the more global picture of the context in which the disease is progressing in the last decades. And then we'll finish with a few summary points at the end. So the aims of the work I'm showing to you today is based on specific qualities of metabolomics and of the metabolome. We're using metabolomics as a starting point because the metabolome is a crossroad where different streams of information meet. The metabolome, of course, is influenced by our genome and the proteins that are encoded in our genome that are the classical way that we know we can influence metabolite levels. But in the last decades, we've become aware of the effects of the exposome as well. So the exposome is a global term that encompasses things like the pathogens we're exposed to, different things in our environments, for example, chemicals, the lifestyle that we have. So that can be how much we sleep, how much stress we have in our life, different things that really can influence our biology and our physiology. The medication we take, of course, and the food we eat are direct factors that will influence the metabolite levels in our body. And this last point, the food we eat, is, as we know now, deeply influenced by our microbiome, especially in the case of food, our intestinal or gut microbiome, that will sometimes provide us with nutrients that we can't otherwise have, but that can also take some of those nutrients away from us. So all of this combined, our genome, our exposome, and this microbiome effects are really converging onto the metabolome. And for that reason, metabolomics is a great functional readout of the phenotype that is the consequence of these different influences. So that's why I wanted to use metabolomics as a starting point and to study the mechanisms that are involved in depression. This is how depression starts and how it looks after several years of growing, because it's a disease that can take a toll for several years. So we will see in this presentation the result of a literature search where I searched these specifically studies dealing with humans and their metabolic levels in depression compared to controls or under the effects of different medications also against depression, exclusively human studies. And we will see in detail 3 of them. Of course, there's a lot more, but I focused on 3 of them specifically because they each provide us a specific aspect that contributes a big part of the final picture I will show you. And then from time to time, I'll make a stop to explain a bit of the biology, because we're not all necessarily aware of all the details of each pathway that comes up in metabolomic result. And then we'll see this final picture in the more global context towards the end. But first, let's have a little overview of what major depressive disorder, this is the clinical term for depression, is. In popular culture, it's also referred to as the black dog. But you're probably aware of the fact that depression is a leading cause of disability worldwide. I won't get into the detail of the symptoms, but the diagnosis, you may or not know this, is still based on questionnaires where the patients make a self-assessment of their mental health. And the pathophysiology is quite difficult to pinpoint, so we're lacking, for example, biomarkers to make this diagnosis, and also actionable stratification biomarkers to help in suggesting the right medication for each patient. And this is clearly visible when you see the responsiveness of patients to first-line treatments to depression, where depending on the study, we still have between 30 and 50% of patients that will not respond to this first line of medication that can have quite large side effects. And so we could use these better stratification markers. The data that we'll present to you will give us an idea of what depression looks like at the metabolic level. For those of you who can't wait, here's a little spoiler. So we'll see a bit of nitric oxide and related metabolites that are part of the urea cycle. We'll also talk about ADMA, or asymmetric dimethylarginine, and we'll talk about signaling related to inflammation, like the kynurenine pathway. And then afterwards, we'll look into the effects of treatments against depression on these different metabolic pathways. So the first paper I want to discuss is a paper from a couple of years ago, and I chose this one specifically because it's the one with the largest cohort that was studied with metabolomics in the context of depression. So we have here a cohort of 460 patients with depression that were compared to almost 900 healthy controls using metabolomics measured in fasting plasma samples. So what the authors could see here, using a panel of almost 200 metabolites, is that some of the interesting findings were changes in metabolites related to nitric oxide synthesis and urea cycle. Namely, you see in the table on the left, we have an increase in depression in arginine and a decrease in citrulline. These two metabolites are used to calculate a ratio, citrulline divided by arginine, that is commonly used as an indicator of NOS activity. NOS is nitric oxide synthase. It's this enzyme that takes arginine as a substrate and produces citrulline and nitric oxide. Nitric oxide being a small molecule that is a very potent signaling molecule for very diverse functions. And you can see in the box plot here that using this ratio, they could see this difference between patients with depression. And I forgot to mention, but these depression patients are having a major depressive episode at the time of sampling. So they're really at the apex of the symptomatic aspect of the disease. And so this ratio is decreased compared to the controls. This is consistent with other research where NOS activity is measured differently. For example, here with radioactive arginine that was used in isolated platelets to measure NOS activity directly. And here you see that the platelets from the patients with depression had lower NOS activity compared to controls. So this is consistent with some of the literature that shows a decrease in nitric oxide production in depression. What's also very interesting, besides the increase in ornithine, which is a metabolite related to the other two through the urea cycle, that has to do with nitrogen detoxification, we also see that ADMA is increased. Now, ADMA, you may not know this metabolite, and I will give you a bit of information about it in the next couple of slides, but ADMA is known to have one function, at least in the body, is that it inhibits NOS. So we have both a sign of decreased activity and an increase in a quite potent inhibitor of the enzyme. So most probably we do have this effect on nitric oxide synthesis in depression. And this was the main finding of this paper on this large cohort of patients with depression. So let's make a little stop to look into the biology. Nitric oxide synthase, or NOS, is present in humans and in mammals in three main isoforms. There's a neuronal form, an endothelial form, and an inducible form. So on top here, we have the neuronal form, which of course is expressed in the brain. And in neurons, it's coupled to the NMDA receptor, which is sensitive to different ligands and will activate through calcium signaling the activation of NOS and help produce nitric oxide. Each one of these NOS isoforms takes arginine as a substrate and produces citrulline and nitric oxide as products. They have the same activity, but the signaling, the activation and the expression of the proteins are different. So in the brain, what this does is it helps with synaptic plasticity, long-term potentiation, and also the regulation of the vascular tone around the brain. In the endothelial form is found in endothelial tissue, and it's famous for the role of nitric oxide in vascular relaxation, also helps protecting the heart and the vessels around the heart, and it's also involved in platelet aggregation. The last form is the inducible form. This one is a bit special because the expression of the enzyme itself is dependent on activation through gene expression. This is classically done, for example, during inflammation by cytokines. And this inducible NOS is particularly expressed in the liver and in immune cells and contributes, for example, to host defense. So this is to give you a bit of background about nitric oxide and the enzyme that helps produce it. The other metabolite of interest in this first study was ADMA, or asymmetric dimethylarginine. Here we see how ADMA is produced. So it's a metabolite of arginine, but the arginine is not methylated like this. It has to be incorporated in proteins, for example, in histones. It's one of the many amino acids that will constitute the sequence of this protein. And under certain circumstances, the arginines will be methylated. This is useful in these proteins, especially if they interact with nucleic acids. So histones classically interact with DNA, for example, but there are others that also interact with RNA, and the methylation helps make a spacer and regulate the interaction between the protein and the nucleic acid. So this is a very useful system, but once the protein has done its job, like any protein, it will go to protein degradation in the proteasome. And there, what we get is not arginine as an individual amino acid, but the methylated form. And this is how we end up with ADMA, with the asymmetric dimethylarginine in our system and in our blood. Now, as I mentioned before, ADMA is known to be a competitive inhibitor of NOS, so it will compete with arginine for the binding site in nitric oxide synthase, but it will not allow the production of nitric oxide. So we don't want to accumulate too much of this, otherwise this could cause problems. And about 20% of ADMA is excreted through the kidneys, but the rest of the 80% is normally degraded by an enzyme called DDAH. That's an enzyme that is expressed or not expressed depending on different signals. For example, if you give the right ligand to nuclear receptors like FXR, you can increase the expression of DDAH and increase the degradation of ADMA. But if you have circumstances where you have oxidative stress, this would rather inhibit the expression of the protein and would then allow ADMA to accumulate. So this is our little summary about these interesting metabolites that we saw in the profile of depression in Metabolomics. Now, in the next couple of papers, we're going to look at the effect of different medication on this metabolic profile. So let's start with this first paper that looks at the first line treatment, SSRIs. So that stands for selective serotonin reuptake inhibitors. This is the first line treatment for depression. Molecules that exist for decades and that have been useful for many patients, but these are also the ones that only are effective in about 50 to 70% of the patients. So here is also metabolomics measured in plasma samples from 136 patients with depression, no controls. And what is done here is that what's compared is the metabolome of the patients after 8 weeks of treatment compared to their baseline before treatment. The result is, first, in the panel of lipids, what could be seen is that there was a significant decrease in several long-chain acylcarnitines. You can see this in the first column of results here on the figure, the brown column, and all these blue squares correspond to long-chain acylcarnitines, most of them. Acylcarnitines, you probably know, are the form under which fatty acids are transported into the mitochondria for beta oxidation. What was also measured were sphingomyelins and lysophosphatidylcholines that did not change very much under the effect of this treatment. But then the next two columns, the phosphatidylcholines, there were quite a few that were increased by the treatment. The panel that was used in this paper also included small molecules, and here we find our usual suspect now, the one that we were expecting from the first study. And what can be seen here is an increase in arginine and citrulline and a decrease in ornithine. Now, if you remember the table from the first paper, there citrulline was decreased and ornithine was increased. So these changes here would go towards putting things back towards the control situation. But arginine was already increased in the first paper with the profile of MDD. And here we see that arginine is even further increased. What does this mean? This becomes very interesting when you see that the levels of ADMA that were also measured were not changed. And what that does is that then the ratio of ADMA divided by arginine is, of course, decreased. This is very interesting because the competition that happens between these two molecules for nitric oxide is an allosteric competition. So the one that comes in at the highest concentration can win the race. And this could be one of the reasons why these treatments have a positive effect on patients, is that they would allow the reactivation of nitric oxide synthesis through an override of the ADMA inhibition by higher levels of arginine. So this was a very interesting finding, even though, let's remember, the ADMA levels were not changed. So we still have these high levels of circulating ADMA in the system. Now, the last paper I wanted to discuss today has to do with ketamine, which is a more experimental type of treatment against MDD, against depression. Ketamine, you might know it as a tranquilizer that is used for large animals or also as a recreational drug. But ketamine has been shown to have positive effects in people with depression at very low concentrations and for short periods of time. So it alleviates some of the symptoms of depression at very small concentrations. So for this reason, it's been investigated for the last few years, both to understand how it works and to see if we can make similar molecules to use as treatment for depression. Now, the findings here are interested. So it's a smaller cohort. It's 29 people with depression. And the graph that I selected here is not one comparing depression to control. It's one comparing depressive patients who were responsive to the treatments to those who were not responsive to the treatment. So here we have these 29 patients. They were all not responsive to SSRIs. And then out of them, 1/3 responded to ketamine. And the differences between this 1/3 and the other 2/3, you can see here, is that the responders had a higher level of one single metabolite, that was arginine, and then lower levels of 15 to 16 phosphatidylcholines, the metabolite kynurenine, and then a reduced kynurenine to tryptophan ratio. What does this mean? So the increase in arginine, we're starting to get used to it. So this could be a very helpful thing to restart nitric oxide synthesis. So that makes sense that they would respond a bit better if they can do this. But then the kynurenine, I will explain a bit more of the biology of it in the next slide. Kynurenine, the ratio of kintrip, and also phosphatidylcholines are typical signs of inflammation in the system. Although here we see phosphatidylcholines decreased, which could be, we're looking here at 4 hours of treatment. So it's relatively quick for changes in levels of complex lipids. So this could be like leftovers of a higher inflammation in these responders before the treatment, and then a quick effect on kynurenine to try and decrease the signaling that maybe will have longer effects later. And to finish with the results in this paper, you see here a little drawing that I made of a very simplified urea cycle with arginine, citrulline, and ornithine. And I drew this just to exemplify a ratio that is calculated that is called the global arginine bioavailability ratio, where you divide arginine by citrulline and ornithine. That is an indicator that corresponds to urea cycle activity. And here, this ratio was increased by the treatment in the responders. So let's look a bit more into kynurenine pathway and also the effects it has in the global context of nitric oxide synthesis. First, on the left here, you see a representation of the kynurenine pathway. And so if you're not familiar with this pathway, it starts with the commitment of tryptophan being converted to kynurenine. This is something that can be done by enzymes expressed in the liver or in immune cells mostly. And once kynurenine has been formed, then we can go to a lot of different metabolites. Two of them are particularly interesting for us because one of them, kynurenic acid, is considered neuroprotective, and the other one, quinolinic acid, is neurotoxic. Very interestingly for us, they both have an activity on the NMDA receptor. So kynurenic acid, the protective one, is an inhibitor, while the neurotoxic one, quinolinic acid, is an activator. So that means one will allow and one will stop the calcium signaling to NOS, and they both will have an effect on nitric oxide synthesis. Now, what we saw with ketamine is it decreases this ratio, so it most likely stops this activation or this commitment of tryptophan to the kynurenine pathway. And what's also known from the literature is that ketamine has a direct effect in blocking the calcium signaling, the calcium channel that does the signaling here. So it really stops any excessive production of nitric oxide through at least two mechanisms here. Now, so we've seen a bit what's available in the literature. Now I want to put this in a bigger picture. So first to connect these little pieces together into one picture, but also to connect them to the greater picture of the context of depression in society and what could be the onset of this disease and what could be factors that promote this disease happening. So to begin with this, I want to talk about two specific points. One of them is the global burden of depression in the world. We all know this is a very prevalent disease, but what you may not know is that in the last 30 years, so since the 1990s, there's been an increase in 50% in depression in the world. This is a huge increase for a disease that is still quite difficult to treat. And in contrast to this, it's really interesting to see that there is one thing that has been shown to decrease symptoms of depression, and it's fasting. So fasting, which is the action of just not eating for a certain time, can turn this black dog of depression into a little puppy. And when you put these two facts together, This makes you wonder, because of course you can't go fasting forever. I mean, you can do that for a certain period of time, but at some point you have to eat something. But it shows us that diet really has an impact. And so could it be that the diet that we've had in the last 30 years, which, as you probably all know, has progressively but surely been converging onto high fat, high sugar, highly processed foods, could it be that this diet has contributed to this huge increase in the prevalence of depression in the last decades? So with this in mind, we started putting the pieces together to see what the picture of depression looks like from a metabolomic point of view. Here we start with what I think is an early event. We know that depression often comes with inflammation. This is something that you find in many studies. So the activation of the kynurenine pathway that we see is something that could come from early on, even maybe before the symptoms of depression. And of course, in metabolomics, words that would translate into activation of the kynurenine pathway, which will influence the NMDA receptor and NOS activity, and can increase nitric oxide synthesis for some time. And in the brain, this could lead us to higher levels of nitric oxide for some time, which we get used to. And we get used to working at a really high pace with nitric oxide in the system. But there's another thing that's happening over time. It's this buildup of ADMA. And ADMA we've seen is an inhibitor of NOS. So now we have something that stops this machine, and the inflammation is still there, but we cannot produce the nitric oxide anymore. And this also, in turn, in organs like the liver, can also disturb the urea cycle. So we start to have an imbalance in the system, especially from the action of ADMA. Now we've seen what the effects of the two medications we studied had on this. So the SSRIs will not impact ADMA, but will change the ratio of ADMA to arginine by increasing arginine. And so this can help arginine to win the competition against ADMA and restore some of the nitric oxide production. And on the ketamine side, we saw we can dampen this signaling of inflammation and also the connection between NDMA receptor and NOS, and also increase arginine as is done by the other treatment, and also increase urea cycle. So it's hitting a lot of points, but no one is really dealing with ADMA. And also, what is the reason for this inflammation in the first place? These are 2 points that the metabolomics doesn't tell us directly, but that I want to try and address by putting depression in the context of diet particularly. So this is what we do here. First, I will talk a bit about Western style diet. So this diet that we've all progressively been accustomed to, with high fat, high sugar, the impacts it has on the metabolome, on the microbiome, and then connect that to our picture of depression at the metabolic level. So one characteristic of the Western style diet is the presence of long-chain saturated fatty acids and an excess of it, which leads, through a disturbance of cell membranes, leads to low-grade chronic inflammation. Another thing that happens with Western style diet is that you influence the microbiome, and you usually see an increase in certain amino acids like branched chain amino acids and ketogenic amino acids that, together with the saturated fatty acids, really push the systems towards insulin resistance, hyperinsulinemia, and all of this also converges onto inflammation, low-grade inflammation. So we have here a sort of little incubator that every time we eat is producing a little bit more inflammation and sustaining the fire of this inflammation every day. But we have systems to deal with this, and inflammation signaling that is telling different systems to cope with this little fire that is in us all the time. And the kynurenine pathway we saw in the brain specifically will also have effects on NMDA receptor and likely cause a slight increase in nitric oxide that is sustained, but that is also manageable for some time. But how does ADMA come into this picture? One other change that happens with a Western style diet is that we see the balance between primary and secondary bile acids shift in favor of secondary bile acids that are much more produced in the Western style diet. What happens with this is that secondary bile acids are very good at disturbing mitochondrial membranes, and they are very good at causing mitochondrial dysfunction. One large consequence of this is the production of reactive oxygen species, or oxidative stress, which we've talked about this once in this presentation. Oxidative stress is the perfect way to inhibit DDAH. We also have ways to activate DDAH, that's FXR. But FXR, which would normally be activated by primary bile acids, is inhibited both directly and indirectly by secondary bile acids. So we can't activate DDAH, and it's even further inhibited by the oxidative stress. So all of this converges onto an environment where ADMA has to accumulate because the kidneys cannot excrete all of it. And so we end up with our inhibitor of nitric oxide synthesis in the system in high concentrations. Following from this, when this impacts nitric oxide synthesis in the liver, this can also indirectly impact the urea cycle, which explains the decrease in urea cycle activity in depression, and also would then interfere with the excretion of nitrogen, or the detoxification of nitrogen from the system. So this was the picture that we could put together from this metabolic results put in the global context. But if you've seen maybe some of our previous talks, for example, on Alzheimer's, you know that this is something that can happen in other diseases too. And we believe that there are multiple complex diseases that have this common origin in our diet. This you can see, we know that the nutrition and the changes in microbiota cause progressively the establishment of metabolic syndrome at different stages. And this causes more and more of the population to have insulin resistance, that then causes different forms of intracellular malnutrition, where the cells don't get the nutrients that they should get. You also get mitochondrial dysfunction, like we saw clearly here, that can cause reactive oxygen species and oxidative stress, and also that cannot function, for example, for energy metabolism as well as they should be. And then you also have hyperinsulinemia, that is a great source of inflammation. And all of this can contribute to different disease types like cardiovascular, neurological, different types of cancers and diabetes. So I showed you today how to do this work of taking the metabolomics to build back to the pathology of the disease. And I think this is something that can be done in many different contexts. And I encourage everyone to try and do this as well. These are the main papers that were used and discussed in this presentation. There's a lot more than came into preparing this, especially the ones comparing depression patients to controls. I chose here a single one that was the one with the largest cohort, but there are other ones, most of them agreeing with the main results. But I couldn't show everything today in this short presentation. And so to summarize, what did we learn? Let's go through quickly what we saw today. So using metabolomics, we could find some findings that were already known about depression. For example, the effect on nitric oxide synthesis, the effect on urea cycle. These are things that are often described in depression research. But we could add to that the increased ADMA, the effect on inflammation and the impact on the kynurenine pathway and on lipids, and connect all these things together. And this is the interesting thing here, is to take these blocks and to connect them together to see which story comes out. In particular, once we had made our hypothesis about how depression works at the metabolic level, or at least parts of it, then we could overlay that with the effects of the drugs that were also studied with metabolomics. And here it became really apparent that ADMA, which has a strong effect in this story, is not addressed by the current drugs, and could really be used as a potential drug target to try and treat the patients that currently don't respond to the medication that is on the market. Another type of connection that we made is to connect the disease into the greater context of society and how the patients evolved in society. We linked the findings that we made to diet, and we could see how this could really help us explain this increasing burden of depression at the global level, and maybe help us to find better ways of helping patients with this disease. Now, this is a lot of heavy stuff, especially if you empathize a bit with the patients. And I wanted to end on a slightly funner note by encouraging us all to tame the black dog, and putting here a picture of my own black dog. There are lots of people who live with this black dog every day, and through medication or other means, I think we should really do our best to help the patients. And I think that understanding the biology better and understanding the metabolome better is a great way to help us progress in the right direction for this. So I finish by saying thank you, of course. Thank you for attending. And also, if you liked The approach that I took in this presentation, I encourage you to connect with me on LinkedIn because I'm very active there and I share a lot of content about biology, metabolites, how metabolites function, how they help with health and disease. And also, if you're interested in podcasts, I'm hosting a podcast that will be launched very soon. That's called the Metabolomist. So you can sign up also to be informed as soon as we release the first episode, if that's something you're interested in. Thank you very much for listening.